So it's my pleasure to introduce Ulzee, who is a PhD student who is jointly advised by myself and Ran Halpern. And Ulzee is going to tell us about his new deep learning-based imputation method. Take it away, Ulzee.
Great. Okay, awesome. Thank you, Sharon, for the introduction, and thanks, Giovanni. That was a very interesting talk. And again, thanks everyone for coming and stopping by. Okay, so I'm excited to share our work on using deep learning for phenotype imputation, and we're calling this method AutoComplete. If anyone's aware of any naming conflicts in general, just feel free to let me know either after the talk. Yeah. Okay, so I'd like to briefly motivate the problem of imputation. So as data scientists, we often find ourselves in scenarios like this. So suppose you're interested in this data set called the UK Biobank. It's a data set of 500,000 individuals. And suppose you develop some sort of method, or you have some sort of insight into specifically alcohol addiction, and you like to try some sort of analysis on this data set. So you might start drilling into this sort of data set, and you might find that your method works best for groups of some sort of subgroup. So there might be a subgroup like unrelated British individuals, which is smaller than the larger number of individuals. And then it actually turns out that the phenotype that you're interested in has only been recorded for maybe a few thousand people. And just to reiterate, so this is saying that for over 99% of the individuals in this data set, this particular feature is missing. Yeah, so this sort of scenario motivates imputation methods. And so what's kind of the setting? So data can be missing in a lot of different sorts of data sets that we're interested in. So why would this be the case? So it turns out, so some features can be costly to acquire. So they could be the product of some sort of invasive procedure, or it could be things like medical imaging, which takes a lot of effort to acquire. There could be some sort of bias in the study that results in missing data. And this is not necessarily a bad thing. It's just saying there's some sort of structure in how the data is acquired. And in terms of things like surveys or questionnaires, you could imagine questions that appear together and et cetera. But just from a top-down perspective, these sorts of combinations of reasons might not be so obvious. And in general, the study that you're interested in might be relatively new. The feature might be still being developed in some way. And oftentimes the types of features that people are interested in are the new ones. And ironically, they could be some of the features that are most missing in your data set. So this kind of motivates imputation methods. And in general, imputation methods try to leverage missingness patterns in the data because for some of the reasons previously discussed, data is not missing at random. And if our method could learn something, learn any combination of missingness or observation patterns from our data, that would be ideal. And ultimately, we'd like to make the best guess possible for these missing values, because otherwise we could be leaving some downstream analytical power on the table. And so, yeah, and obviously the end goal is it would be great if by doing imputation, this improves our downstream analysis. So in particular, we were interested in doing imputation for the UK Biobank, and especially phenotypes in the UK Biobank. So the UK Biobank is a very large data set of almost 500,000 individuals. And in this data set, so it contains genetic information for almost the majority of the people. But it turns out things like phenotypes, so peripheral information, any sort of demographic information, and there's also imaging data as well. And so any of these types of data could be highly missing. And so even though you have all this genetic information, depending on the analysis you might want to do, you could be limited based on how missing some of the features you're interested in. And so in particular, we were looking around our peers, what our peers were working on, and some of our collaborators were working on. And we gathered two types of data sets, two collections of phenotypes. So one in particular was related to cardiovascular types of information. So things that come from blood labs, your lung capacity, phenotypes related to that. And we also gathered a group of phenotypes related to depression. And there's many ongoing studies on depression. And many people are trying to understand depression as a disease better. But a lot of facets of depression are still not very well understood. And so we were very interested in seeing if we could do imputation for this data set involving depression. And I just wanted to highlight that for either of these data sets, some of the features could be very highly missing. So we see 90% of some of the features are missing, up to 99% of some of these features are missing. Yeah, so from here, I'd like to share, I guess, more of a computational story. From the onset, we realized that whatever imputation method we wanted to try, that this has to scale to hundreds of thousands of individuals. And this was immediately a challenge for some existing methods that we found. We wanted to do imputation for both continuous and binary format features. And we also found that handling this sort of heterogeneous data types could be a challenge for some existing methods. And also we wanted to find realistic ways to measure accuracy. And especially we wanted to focus on measuring R-squared accuracy. And this is because R-squared is one way to measure, it's a measure between 0 and 1. And it's a one way to measure the percentage of variability in the data that can be explained by any of these tested models. And in particular, just to revisit the topic of computation. So we tested, we're illustrating two methods here. So there was one method, Softlmpute, that was very prevalently used, and also mice, which was another imputation method. And we found that these imputation methods could scale very, very differently depending on the size of your data. Yeah, so given these considerations, we developed this method called AutoComplete. And so this is a deep learning approach to doing imputation, and it uses an architecture called autoencoder. And the implementation for these methods could be a little more involved, but the intuitions could be pretty straightforward. So I just wanted to illustrate the intuition here. So suppose you have some feature vector X that has P, that's P in dimension, and you have some function X that takes the feature vector and can roughly reconstruct its original input. And what this H does is it takes X and then it projects it to a lower dimensional representation. And then through some sort of either inverse or transpose transformation, it brings it back to approximate reconstruction of its input. And in terms of something like constraint optimization, you can just define some sort of reconstruction objective in terms of the reconstructed version of X in its original form. And the idea of autoencoders is that we're basically saying this lower dimensional projection, this function H, so instead of using some sort of projection matrix, we can use deep neural nets. And so deep neural nets are nonlinear, but they're also differentiable. So another benefit of this is we can play around with a lot of settings and also define our own custom objective function. Yeah, and so one requirement that we wanted to make sure our method could satisfy is being able to handle both continuous and binary phenotypes. And the way we did that is to define a joint objective for these two types of data formats. And so just to quickly illustrate that, so if we partition, for simplicity, if we partition the first half of our input vector to be continuous and the second to be binary with some index j hat. So we defined, we could kind of flexibly define an objective function for the continuous phenotypes. And then for the second half, we defined an objective for the binary phenotypes. And we could combine these two terms to form an objective. Okay, so we have our autoencoder, and I guess now we'd like to know how can we do imputation using an autoencoder. And one thing about autoencoders is that if you can fit an autoencoder on your data, if you can find some sort of low dimensional representation of your data, then there's benefits to that. So in some ways, that means you can, there could be, depending on slight perturbations in your input, you might still be able to reconstruct the original data. And so we wanted to leverage that sort of feature of autoencoders. And the way we induce imputation using autoencoders is that we wanted to simulate missingness and force our autoencoder to reconstruct these simulated missing values as if they were observed. But to do this, we wanted to make sure our simulation procedure is somewhat realistic. So we developed a method that we're simply calling copy masking, but the idea is very simple. So we take existing missingness patterns in our data and we propagate it to from a set of individuals to another set of randomly selected individuals. And this is randomly done. And the idea is that we want to propagate any sort of non-trivial missingness patterns that are already in the data. And we could keep running this sort of copy masking procedure. And so we could create settings of missingness, starting from 1%, where the simulated missingness is very small, all the way to 50%, where that would be like an extreme case. And actually, during the training procedure of our autoencoder, we add additional augmentation. So essentially, we do both copy masking and additional random masking in our training. And empirically, we actually found that this leads to our autoencoder being more robust. And so the idea here is also very straightforward, but in addition to some level of copy masking, we want to induce just a randomly uniform level of missingness in our data. And so in combination, so we have both copy masking plus random masking during training, and we're asking our autoencoder to reconstruct these missing values as if they were observed. Yeah, so we were actually very encouraged by our early results because we found that this sort of autoencoder implementation came very close to some of the existing methods. For example, some of our initial tests, the autoencoder was very neck and neck with Softlmpute, and we found that overall Softlmpute was one of the best performing imputation methods. And so we drew very close comparisons with Softlmpute. So these are some of the figures that we were looking at when we wanted to measure overall performance of these methods. We're looking at R-squared accuracy between simulated missing values and their imputed missing values by using each of these methods. And these R-squared accuracies were averaged over all the phenotypes. So for the blood labs phenotypes, there were about 80 or so, and there were about 370 phenotypes for the depression data set. So we averaged all these accuracies to assess some sort of overall performance. And some of the patterns that we saw was by sort of carefully tuning this autocomplete architecture, we were able to get it to perform relatively neck and neck with Softlmpute, and even better for some phenotypes. And so that kind of contributed to its overall R-squared accuracy being slightly higher than Softlmpute. And also, we saw that R-squared accuracy was relatively more robust as we induced additional missingness. So as the simulated missingness percentage increases from 1% to 5% and so on, we saw that autocomplete might be most robust in settings of additional missingness. And the methods that we compared to, they all degraded pretty quickly with these additional, in these additional missingness levels. And just to reiterate one point about R-squared accuracy, for these very large populations, even small gains in R-squared accuracy indicates a higher percentage of the data being explained by the model. So even any small increases in accuracy were favorable. So we drew a much closer comparison between AutoComplete and Softlmpute, as we saw that Softlmpute was one of the best performing imputation methods, and it was basically the next best performing method in our study. And so basically, examining these accuracies closely, we saw that for one encouraging thing was that for many of the phenotypes, so both methods seem to relatively agree. And on top of that, from using autocomplete, we saw that for a few select phenotypes that these improvements in accuracy would be significantly, they would be significant. And yeah, just to point out some of these increases in accuracy. So we saw that, for example, for some depression-related phenotypes, that there were phenotypes that were not very well imputed using Softlmpute. But for them, we saw that autocomplete could would improve very well because it's able to achieve some sort of accuracy, and so on. Yeah. Yeah, again, so considering these large populations, any sort of even incremental increases in R-squared accuracy and for any other metric could be very impactful. So we looked at, we measured relative improvements between other methods and AutoComplete. And we saw that the story was mostly consistent. So Softlmpute was, again, the next best performing method. And in terms of R-squared, we saw in some of the earlier settings that Softlmpute and AutoComplete were comparable. But again, there were a select group of phenotypes for which AutoComplete seemed to improve very significantly. And in terms of comparing with additional methods, so we compared with high VAE, which is a variational autoencoder. So it's a probabilistic neural net-based deep learning approach. There was GAIN, which was generative adversarial. So it's another discriminative deep learning approach. And compared to these two deep learning approaches, it seemed like using AutoComplete would be much favorable for any setting, just in general. And also we compared with nearest neighbors imputation, which is available for many software data analytics packages. And we saw that in comparison to KNN as well, the relative improvements would be quite significant. And revisiting one of the points from earlier, so we wanted to make sure that this method is scalable to the scale of the UK Biobank. And since our approach is based on deep learning and it's an iteratively fit model, we essentially saw that our method was easily scalable to the size of the UKBB. And so running this, so fitting this model for 10 epochs, the running times were very similar with Softlmpute. And we could try to make this model more robust by running it for additional epochs. And even then, so for, let's say 100 epochs, we saw that fitting this model on the UKBB would still be very realistic. So just to summarize, so we developed a deep learning based imputation approach called AutoComplete. We used an autoencoder model to do this. And we tested this approach on 2 cohorts of phenotypes from the UK Biobank. And we'd like to see if our method generalizes to additional types of data sets. So if anyone has any data sets that could benefit greatly from imputation, I'd actually be very happy to connect with you, and maybe we could work together to see if AutoComplete could help. We also did our best to do realistic simulations to measure realistic accuracy. And so we implemented a method called copy masking. So overall, we saw that AutoComplete was mostly favorable for imputation in our simulated ranges from 1% to 50% of missingness for these UK Biobank phenotypes. In particular, so we were interested in these depression-related phenotypes. And we saw that for several of these phenotypes that the improvement in R-squared would be quite significant. And also for this secondary data set of cardiovascular blood lab related phenotypes, we also saw quite significant improvements in imputation accuracy. And again, these comparisons, we did the most detailed comparisons at 1% of simulated missingness because that would be where the simulated data is closest to the real data. But with simulating additional missingness, we saw that AutoComplete would actually be much more robust than the methods that we compared to. Yeah, so thanks for listening. I'd like to thank Nasai and Andy Dahl, who provided the depression-related data set. Also, Ali, who helped with the initial steps of the study, and my advisors here on. And I'd also like to thank the countless collaborators who helped give feedback to developing this method. So thank you.